This is Module A4, Muscle Tissues, and this is Part 2 of Objective 4. And again, in Objective 4, we're looking at the characteristics of the muscle fiber as well as the sarcomere. So in Part 1, we were looking at some of the anatomy of the muscle fiber itself. Let's just kind of review that information. Up here in this larger figure, we find that back it up here. We can identify this as a muscle fiber because we find the multinucleated cell. And from that, we can also identify the sarcolemma as being the plasma membrane of the muscle fiber, as well as the sarcoplasm, which would be the cytoplasm, all of this region here. We should also be able to identify this little organelle as being the mitochondria. We would anticipate that muscle fibers utilize a tremendous amount of ATP, and we would expect to find a large amount of mitochondria to help supply that energy. And moving down into the figure at the bottom of the screen, again, we have some more detail. We find these little small channel entrances to the T-tubules, and then we see that the T-tubules are also running down, wrapping around all of the myofibrils. We have the sarcoplasmic reticulum, which is all of this blue organelle. Remember, that's closely related to the endoplasmic reticulum, and its main function is to sequester or to store and concentrate calcium ions. Remember that the sarcoplasmic reticulum produces these large pocket-like structures that are known as terminal cisterna, and two terminal cisterna and one T-tubule give rise to a triad. Okay, these three structures are going to give rise to a triad. Looking at the myofibrils, we find that there are two protein types, and you can actually see them here. Here we have a purple protein. This is the thick filament. Remember, the thick filament is myosin. And the thin filament is actin. Okay, so what we're going to start moving into is the specifics of this region of the myofibrils, and the functional unit of that is called the sarcomere. So sarcomeres are the contractile units of the muscle. This is the unit that undergoes or allows for the contraction, and each muscle fiber is composed of dozens and dozens of sarcomeres. And it's these sarcomeres that actually give muscle, skeletal muscle, its striations. Up here we have, in the upper figure, we have a histological prep of skeletal muscle, and if you remember from lab, skeletal muscle is identified by a few characters. One is its striated appearance. Cardiac muscle is also striated, so the way we differentiate skeletal muscle is we look at both the placement of the nuclei. We know that in skeletal muscle, the nuclei, it's multinucleated, and they're pushed towards the periphery of the cell, so they're going to be found largely on the periphery of the cell. And the second characteristic for skeletal muscle is that it does not branch as frequently as the cardiac muscle. So these are very long, straight fibers. There's no branching. And again, that striation is caused by the placement of these proteins, these myofilaments. And if we look at this is some type of electron microscopy, we blew up the muscle slide histology prep, and we actually see the alternating dark, light, dark, light, and that is what gives rise to the striations that we see. With that, with those structures, with that repeating pattern, we can identify a number of bands and a number of lines. And of course, these have all been given names by anatomists. So let's start out with the A band. The A band is the region, the dark band region. So here we have the A band, and it's obvious that it's relatively dark in color. And along with that, we see right in the middle of the A band, we have a dark line that is known as the M line. And there's really no easy way to learn this material. You just have to commit it to memory. The I bands are the light regions, the light bands. And in the middle of the I bands, again, we're going to find a very dark line known as the Z line. Other structures that we can identify, we have an area known as the zone of overlap. And the zone of overlap is an area where the thick and the thin proteins overlap. And this creates the darkest area on the light micrograph. So if we were to try to identify the zone of overlap, we would see it right here. This is the darkest region within the A band. We see that as we approach the M line, we do have some lightening or some lighter colored regions. So the zone of overlap is going to be the darkest. And again, this is where the protein filaments are overlapping. And after we identify the zone of overlap, we have a small region that actually contains the M line that is lighter in color, and this is referred to as the H band. The H band is comprised largely or completely of thick filaments. We do not have any of the thin filaments or actin present within the H band. Other proteins associated with skeletal muscle, one would be titin. These are strands of proteins, and titin stretch between the thick filaments, or the myosin, and the Z line. And we'll look at a figure in the next slide that will help you understand its relationship. But the purpose of the titin is to help stabilize and, well, basically to help stabilize the myosin. So again, here we have a diagrammatic representation of our sarcomere, and we can identify the A band as being the dark band found under microscopy, and the I band being the lighter band. We have the M line and the Z line, and again, all these structures are made up of three proteins that we're going to discuss. We have the actin, the myosin, and the titin. And we had just introduced titin, and here we can see titin as these green curvy lines. And again, what titin does is it holds or stabilizes the thick bands, the myosin, to the Z line. And I really hope that this all comes together when we start looking at muscle contraction, because once you see how these different elements interact, it really makes it easier for you to understand these different parts. So just bear with me. There's a little bit of memorization early on. Hopefully it all comes together a little bit later. This slide, this is figure 10.5 from your text, and it basically looks at a sarcomere, and it divides it, or it takes cross sections of different regions or parts to show how all these protein elements are related. And up to this point, we have yet to define the sarcomere, so we should probably define the sarcomere. The sarcomere runs, it's a region that that is found from Z line to Z line. That is found maybe between Z lines. So here we have one Z line, and over here we have another Z line. The sarcomere is the entire unit between Z lines. Okay? So between this Z line and the next one, we would have another sarcomere. And that's going to continue the entire length of the muscle fiber. And again, this is the contractile unit of the muscle fiber. This is the unit that contracts. When this sarcomere contracts, these two Z lines become closer together. They're going to move in towards the center, towards that M line. So this is the contractile unit of the muscle fiber. So let's kind of look at how muscle is organized, and we can start out with the organ itself, the skeletal muscle. We looked at the skeletal muscle. We know that it's associated with a number of connective tissues, vasculature, nervous supply, and it's composed of a number of muscle fascicles. As we look at the muscle fascicle, again, it has its own connective tissue, blood supply, nervous system input, and it is composed of a number of muscle fibers. The muscle fiber, with its connective tissues and both cardiovascular and nervous inputs, are controlled by, are comprised of a number of myofibrils. The myofibrils, with its close association with the sarcoplasmic reticulum, is going to be composed of a number of sarcomeres. And remember, the sarcomere is the region that extends from Z line to Z line. So this is your sarcomere, the contractile unit. So what is the function of the sarcomere? Well, again, it's the contractile unit, so the function is to undergo contraction. And we know that the sarcomere has a very close association with the organelles that we discussed, such as the T-tubule and the sarcoplasmic reticulum, or SR. What is the function, again, of the transverse tubules? The transverse tubules, they will deliver the action potential or nerve impulse. I'm just going to abbreviate action potential AP, and then it's the nerve signal or nerve impulse. So the transverse tubules will deliver the action potential or nerve impulse ultimately to the muscle fiber. And so how does the cell respond to that nervous impulse? The nervous signal basically causes the release of calcium ion. And it's the interaction between the calcium ion and the proteins, the proteins within the sarcomere that ultimately lead to contraction. Contraction is nothing more than the interaction between the actin and the myosin. And it's the actual structure of these proteins that allows for that interaction to occur. And we have a number of proteins that play a part. I guess the first protein we should look at is the actin. And actin, we can identify separate subunits to actin. We have F-actin, which is the filamentous actin. Down here in this bottom picture, we can actually find the strands that are F-actin. And F-actin will come as two strands will actually form a helix around one another, giving rise to the filamentous actin. So it's two twisted rows of individual globular G-actin. The G-actin are actually all these small spheres, and as they all link together, they create F-actin. There are two other proteins that are closely associated with actin. One is the tropomyosin, and the tropomyosin is this, again, very long double-stranded protein that runs along the F-actin. And its function is to kind of block the interactions between the thick and thin filaments. And then lastly, we have troponin, and troponin is right here, and it's a small protein, globular protein unit that is sitting bound to both the tropomyosin and the G-actin. And this is the protein that reacts to calcium. So when calcium is released, calcium will actually bind the troponin molecule, and that's going to initiate a series of events that lead to muscle contraction. So calcium binds troponin, and that causes the troponin-tropomyosin complex to alter its shape. And in doing so, it's going to expose the active sites on the F-actin. Let me see if I can draw this. I don't know how well this is going to be. So what I'm drawing is your F-actin. Remember that each one of these individual circles represent the G-actin, the globular actin. So here's our F-actin, and I'm simplifying it. There are two strands that are twisted together. I'm just representing one. And all along this F-actin, we have a little active site that's near the top, and I'm just going to color that in on a few of these circles. So this is the active site, and we'll talk more about its activity in a minute. Just imagine that all of these are filled in like this. And sitting on top of the F-actin, let me change my color, is your tropomyosin. And again, this is a very long protein that sits right on top of the F-actin, and it blocks these active sites. It sits right on top of the active sites and blocks them effectively. Associated with the tropomyosin is the troponin. And the troponin is a small protein that binds the tropomyosin. And when calcium is released, when calcium ions enter the picture, they bind the troponin, and this causes a shift, a change. The troponin and tropomyosin, here in red, they shift out of the way. And what that does is it frees, or it opens up this active site on actin. And when that active site is open, it then allows for the interaction between the actin and your myosin, which we'll look at next. if we look at the actual structure of the myosin, let me just fast forward to a figure that shows that. We have a head region and we have a tail. It kind of looks like a golf club. And this head will pivot. We can see here with this arrow that it's representing movement. This head can pivot back and forth. And all these myosins, proteins, they're all bound together with other myosins in this configuration. They look like a whole bunch of golf clubs kind of bound together. And this is basically your thick filament. If you were to simplify this, we see up here that the myosin is all of this purple protein, and this is basically what that structure looks like, slightly blown up, with all these little golf club-like proteins kind of bunched up together. The red up here represents the actin. They represent this picture right here, the actin with the tropomyosin and troponin. So it's through the interaction, and let me just go back here to make sure everybody has this material. Thick filaments contain the myosin subunits, and of course they're associated with titin, which helps to not only stabilize, but it also aids in recoil after the sarcomere is stretched. And then we have the two components of the myosin molecule. You have the head and the tail, with the head being able, or it can pivot. It can actually can change its configuration and move. So during contraction, the myosin heads, the golf club heads, interact with the actin filaments. The actin filaments, once that active site has been made available. And you get the formation of a cross bridge. Okay, your actin, with all of its little active sites, and I'll draw it in a different orientation this time. Once the troponin tropomyosin complex is moved, these active sites are open, and then here you have your thick filament, your myosin, and the head of the club will interact with the active site on the actin. And once you have that interaction, once you have a cross bridge, once these two connect, all you need is for this head to pivot. As that head pivots, it's going to pull the actin in this direction, and that's going to shorten the sarcomere. And we're going to see a really good animation of this activity. So it will really, if my drawings aren't doing it for you, that animation should clear up a lot of what I'm lacking. So again, this is a lot of material. I do understand that you really need to sit down and understand all the anatomy and all the structural units that were discussed. And once you have that memorized, it's going to make the rest of this module considerably easier.